Okay, without further ado, I'm going to present the first keynote speaker of the day, the second of the conference, which is Dr. Vázquez Domínguez. Dr. Aya is a professor at the Institute of Ecology of the UNAM. Her lab uses genetics and genomics to study problems in ecology and evolution. And they study a range of taxa from invertebrates to vertebrates and scales ranging from the local to the regional to the global. She did her bachelor's and her PhD at the UNAM, first in the Facultad de Biología and later at the Institute of Ecology, and did a postdoctoral stay at the University of Queensland in Australia. So, without further ado, podemos ir a la charla que va a estar remota.
Okay, I'm guessing I am in. So I'm just going to go ahead. Hi, good morning, everyone. Good afternoon from where I am. I'm in Spain right now doing a sabbatical. And I'm going to tell you a little bit of natives, hybrids, and invasives in how intriguing genomics we have been doing in our lab. Okie dokie, yeah, I'm just being confirmed that you can hear me. Excellent. Okay, so one of the research lines that we've been working in our lab is trying to figure out how species colonize new environments, which is still a fundamental question in ecology and evolution. And there is a myriad ways of and approaches and how to explore this fundamental question. And for the talk that I'm giving you today, I'm concentrated in biological invasions and hybridization systems, which offer a unique opportunity to explore different things. For example, patterns and processes of species colonizations into novel environments. They help elucidate the roles of evolutionary, biographical, and ecological forces driving species distribution. And of course, they help to monitor biotic interchange. I'm going to tell you about two different stories, stories that have taken a long time, many years. You know how science is. You have a couple of questions and then you have new questions that you have to and want to continue exploring this very fun way of doing science, which we love. And so it's been many years. And the two stories, the leaders of these two stories I'm going to tell you are an amazing couple of PhD students I had in the past, Zaira Espíndola and Gualberto Pacheco. So everything you'll see, it's their PhD work. We'll start with the turtles study, and after these many years, it came to be condensed in these two main publications. There were some others, but these are the main ones. And what I'm going to tell you, it's in there, so you can go ahead and look at if you wish. For the turtles study, there is one slider turtle, Trachemis scripta elegans, which is called the red ear slider turtle or the Japanese turtle, which in the 1950s became a very popular pet for kids. I had one in the 70s. And it was sold it was cultivated in Japan and sold to pet stores around the world. That's why it's also called Japanese turtle. And after not many years, it became it's classified now as one of the 100 worst invasive species in the world. It is absolutely successful invasive in many parts of all the continents in the world. So we were interested in this species, and let me tell you why. And that is because the Trachemis, the slider turtles, there's about 15 species and around 19 subspecies so far, and most of the diversity it's within Mexico. We have many of the species and subspecies. And more importantly and more interesting is that this species, the red slider turtle, its native distribution is along the west coast of the United States, and it passes the border a little bit down to Mexico, into the northern part of the state of Tamaulipas. That is its native range. So we had this species in our territory, shared with many others, but it forms a geographic gradient that shares with other species. One is Cataspila, Trachemista cataspila, which is endemic to Mexico. It's only distributed in Mexico. And then going south along this gradient, we have Venusta, another of the species, that continues along the Gulf of Mexico throughout the Yucatan Peninsula, and it reaches Belize in Central America. And we also have a microendemic, the Trachemista taylori, very, very micro. It's it distributes in a wetland system within a desert area in the north of Mexico. So having these different congeneric species, we wonder, first, what would be the genetics and evolutionary patterns among these trachemists? The phylogeny is well resolved, but we wanted to explore this a little bit further. But we were most interested in how the invasion scenario would look in this region, given the co-distribution of other congeners in Mexico. And I say this because the invasive distribution of these species had been very well studied, except in Mexico. We didn't even know. We suppose it wasn't as invasive as it is everywhere else, but we didn't know. And trachemists are known as good hybrids, good hybridization systems. So we also wanted to know if we would be finding hybridization among these four species. And to do that, we went to the field. We did a very extensive field work. You can see all of this here. You will see the color codes for the invasive, for the Scripta elegans in red, Cataspila in purple, following the gradient, orange for Venusta, and blue for taylori. We had many samples from the widest native range from museum samples. We didn't do field work in the United States. And we were able to analyze 49 localities. This data set is 261 individuals. We had mitochondrial sequences, nuclear sequences, microsatellites, and also genomic data based on more than 63 SNPs. Okay, I'm not going to explain in detail our methods. I'm going to go right into results. But if you later on want to know more, you can, of course, ask me. And you will always see in the bottom of the results I'm showing you what we did, what's the method, and what is the software we use. So for our first question, we found, as expected, that the four species are clearly divergent in accordance with their taxonomy and their natural distribution. And you can see how Scripta elegans is the most recent diverge, and also the highest diversity along its native distribution. Okay, these four trachemists diverged about between 13 to 10 million years ago. It's a very old system. And we did not find signals, any significant signals of hybridization. We again, you can see here the color code, and you can see we have four genetic clusters, clearly differentiated. We identify some ancient integration, especially between elegans and Cataspila. Remember, these are the two neighboring ones. And also identify more recent admixture processes between Cataspila and Venusta. Again, the other two neighboring along the distribution, but no really significant among the four species. We also did not find signals of current contemporary gene flow. Based on this analysis, estimated effective migration surfaces, we identify very low effective migration in comparing the Scripta elegans, the red slider, with both species, congeneric species, Cataspila and Venusta. You can see here in both figures that there are strong barriers to gene flow. indicating no current or contemporary gene flow. So everything is supporting this very highly structured genetic structure between these species. We also had this is very, very intriguing. We expected, as I said, that we would find a lot of individuals of Scripta outside its native range along our field work, our field sampling sites, but we did not. We did not find one individual of Scripta in the natural scenery. You can see here, this is one of our sampling sites, very natural, very wild. And here is, so all of the sampling points that we have for Trachemis scripta along Mexico, along our sampling region, came from artificial water bodies in parks, in middle of the city. You can see this is one of our sampling sites in Jalapa, Veracruz, in a park downtown, and it was filled with turtles from two species which were not native there. They were absolutely exotic, and this comes from people having the little turtle as a pet, and then turtle gets big and they will release it into these artificial ponds. So we didn't find it in the wild, but our samples come from these artificial systems, and this allowed us to compare genetically the Scripta elegans from the native area and the non-native area. So this is our sampling from exotic places, and you can see they're absolutely and significantly differentiated genetically. So the native genetic composition and genomics are different from the introduced ones in Mexico, and this should apply to all over the world. So, and having found all this evidence of no intermixing, how Scripta was invasive in Mexico, we wanted to explore a little bit more what could be the reasons. You can see here how they're very different morphologically. They're very distinguishable in the field, and there are different processes that can be going on here, for example, competition. But one thing that we could actually measure was the ecological niches of the species, to see if they had different ecological variables dominating their habitat, and that could be one way of knowing it's the ecology that could be associated with their non-intermixing and their being very well separated. And we also estimated in a more detailed means, the Scripta elegans fundamental niche. We took all different measures to obtain the fundamental niche and to evaluate if it was related to its invasive success. And what we found is this is based on in evaluating and measuring the niche overlap based on the ecological centroid of the environmental niche between specifically elegans from its native range and its congeners. And you can see how they follow, again, the geographical gradient, and they are differentiated. The overlap between elegans and Cataspila, the one that follows in the gradient, is less than 40%. And this is even smaller when we compare it to Venusta. So their ecological niches are really differentiated, little overlapped. In contrast, see what happens when we did this comparison with another 11 species all over the world, where Scripta elegans has invaded, established, and displaced the natives. And you can see in Western Europe, Asia, Australia, in all those cases, or most cases, the niche overlap is absolutely it's almost 100%. So meaning that the ecological niche for this invasive compared to where it has invaded and displaced, it's almost the same. So it's a different story, as you can see, what's happening with the congeneric species in comparison with the places where it has invaded. So very briefly, what we can say is that elegans and its congeners are genetically differentiated along their natural distribution, and that native and non-native elegans also is genetically differentiated, and that in contrast to most invaded regions, there's little mixture and hybridization, and the invasion is limited along the gradient with this congener. And I'm not going into more detail about the fundamental niche, but we found that actually elegans is occupying a very small subset of the fundamental niche it could inhabit. And this is bad news, because that means worldwide is a wide range of suitable environmental conditions that it could occupy. So it has high potential to invade even more places in the world. So that is not very good news. So I'm finished there with the turtles story, and I'm moving to the crocodile one. Again, many years, and it's been, the product of that is these different publications. And let me tell you very quickly, for those that don't know, we have two crocodile species in Mexico that distribute in Mexico. One is the common American crocodile that you can see has an ample distribution going along the Pacific all the way Central America to northern South America, along the Caribbean. And we have Crocodilus morelletti, Morelletti's crocodile, or in Spanish we call it Cocodrilo de Pantano, which we like to say is quasi-endemic in Mexico because it's restricted to the Gulf of Mexico, the Yucatan Peninsula, and it goes a little bit farther into Central America, in Belize and Guatemala. You can see they are different, very different morphologically. The form of the face is different, the weight, how robust they are, and there's also certain patterns of the scales above the head that are taxonomically characteristic. You can distinguish by the count of scales. And another important difference is that Acutus has salt glands that allow it to process salt water, and that's why this is the species that goes more into the ocean. While Morelletti is absolutely restricted to more freshwater, pantanos, marismas, that kind of habitat, because it doesn't have as well developed those salt glands. So for this, it has been known for a long time that in their historical zone of contact, especially in the northern Yucatan Peninsula, it has been known that they hybridized. Crocodiles, all species, all 24 species are hardly hybrids, faculty of hybridization. So we knew there was historical hybridization in this area, but all previous studies had been very local, like sampling here a little bit, sampling there a little bit. So Gualberto, what he set to do from much before he came into my lab was to sample throughout the entire country as much as he could, a lot for the two species. So this is part of most of the sampling of the study, and that allowed us to have 92 localities, around 370 analyzed. We have more. Also, we had mitochondrial sequences, microsatellites, and genomic data. And the first results that came out with microsatellites were that actually the hybridization zone was much more widespread than what the literature had said, not concentrated only in the northern Yucatan Peninsula, but having a gradient along almost the entire Gulf of Mexico. That was the first very surprising and interesting result. And another one was that the populations of the parental Crocodilus morelletti, we don't like to say pure, but they are non-unmixed parental populations, were few and were very isolated and located, restricted to the northern part of its natural historical distribution. Okay, so that was okay, we have here a problem. Morality is not as well conserved, or as there are not as many populations as they were thought, and this had many implications in conservation, as well as having so many hybrid individuals along this entire gradient. That was another conservation conundrum, because these were thought to be actually Coccolio's morality mainly. So we wanted to go farther into this, and here we found these two sites where we had high signal of hybridization, and so analyzing that data in more detail. And this was undergrad thesis of another student. What we found is that these three populations, one is in a national park, Cañón del Sumidero, where Crocodylus acutus, the American crocodile, is naturally distributed. But there were other two, these other two yellow dots, that had Morelletti individuals. And so you can see El Aguacate is one of the localities, which is all Morelletti. And we had individuals from the native distribution, just to make sure we were we're having the genetics correctly, and yes, we did. And Villaflores, which is down here, had long-term hybrid individuals, F1, F2, and back crosses, meaning that people had been moving intentionally for these crocodile farms. They had been moving Morelletti to these specific areas, and they were having highly hybrid individuals. So that's why we had this high hybridization signal, where we weren't supposed to have Coccolilus morelletti. And so going farther into the analysis, we obtained genomic data, and we indeed found or confirmed that we had 5 lineages, our 2 non-admixed acutus and morelletti, especially morelletti, with very isolated populations. We had 2 lineages of hybrids, and this is really interesting. One that is more this Gulf of Mexico widespread, more recent, and one more ancient, more historical, the original Yucatan Peninsula hybrids. And you can see here also they are separating. And to our surprise, we also identify a Caribbean, non-parental, a non-admixed, a parental Crocodylus acutus lineage in the Caribbean islands that you can see is really distinctive from the Pacific acutus. So this was really, we were so happy with these results in terms of, we didn't expect them, and we wanted to go further and did a whole battery of different analyses to confirm. And yes, we have 2 Crocodylus acutus lineages, one restricted to the Caribbean islands, that is Cozumel and Chinchorro in the Caribbean side of Mexican Caribbean side. We have our 2 lineages, hybrid lineages, with more historical integration, the Yucatan one, and the Gulf of Mexico having still much more admixture from Morelletti. We confirm this further, and we have historical migration data indicating, again, how an ancient acutus lineage had migration into the Gulf of Mexico and Yucatan Peninsula hybrids. So all of this allowed us to conclude that we had this different diversification and different species boundaries, even one that we didn't expect. We also went to do some ecological studies, and just to be very brief, both hybrids lineages have different ecological niches, and that distinguish them from the parentals, and between them also. So this was also very, very interesting to follow. And very recently, this is only data we are still working. This is a working in progress. We are trying to pinpoint local adaptation. We've found diversifying selection, loci that are diversifying different in the Gulf hybrids compared to the 2 parental species. We have found associations between some of these outliers with different environmental variables, mainly temperature. And we have also identified candida loci and annotated some of those. And there's one gene that pops out related to bone formation and morphogenic processes with bone proteins, which can be very, very important because as I told you in the beginning, this bone formation is related, can be related, these genes can be related to the building of the scales that distinguish the two species. So we're further going into that to try to, this is a whole new project that we're working on. And that will be, oh, I went very fast, but I'm on time.
Great. Thank you, Aya. Can you hear me?
See, yes, I can hear you.
Great. So we can have some questions now. We have a question over here.
Hi, yeah, very interesting.
Hi.
I'm thinking it must be a bit hard to sample crocodiles. So I'm guessing how you go about that and what part of the crocodile you sample, and also if given that the complications I assume exist for sampling them, if you have considered like non-invasive sampling methods. I don't know if like they shed scales or skin or
Okay,
poop
okay.
or something.
You know, as I said, this most of this, well, the whole sampling was done by my student and a whole team of crocodile fanatics, or not fanatics, but people that have been working with crocodiles. And actually, they did catch every single individual we have in our database, and they take either blood samples or they mark them with the doing cuts in the scales. So we use scales also for obtaining DNA. So it is hard of course catch them, but there's a lot of people that know how to do this, and it was much more interesting because we have all measures. We have, I didn't mention and I didn't show the results, but we also analyze morphometric data, and they distinguish between the both parental species and the hybrids. And in order to have that kind of data, you have to catch the animals. So yeah, you couldn't use feces, for example, because they're most in the water. So it is important to have the individual.
We have a question over here.
Thank you very much. Yeah, it was a super interesting talk. I'm just double checking if I understood. So the non-endemic Morelletti crocodile lives mainly in sweet water, right?
Yes, in some, as you could see in the map, I had the points more like with in the continent.
Right.
So
right.
there are lagoons where they inhabit. Yes.
Okay. So my question is like maybe whether there is a genetic adaptation for the salt crocodiles to be able to live in salt water, and since you have hybrid data, then you could do sort of like a mixture mapping to trace that possible genetic adaptation to salt
Yeah.
water in the Morelletti crocodiles that now, because they're hybrid, they're living along the Gulf and in the in the Caribbean.
Absolutely.
I'm
Yeah. Yeah.
just wondering if you guys have thought about that then.
Of course. Yeah, that's what we want to do. We know that naturally they have these differences in the salt glands that I mentioned. So acutus would have them very, very well developed, and that's why it had go into saltwater while morality can't. And what we are trying to figure out with the outlier loci and mapping the going to map this into the crocodilus genome is to identify not only these bone genes. We know there are genes related to salt metabolism, and we're asking to see if there are differences. We expect that naturally Morelletti and acutus have differences in those genes, and we want to know how it is in the hybrids. We know from the field that the hybrids are more salty most of them, especially in the northern Yucatan Peninsula. But we are wondering what happens to the more that have more ancestry of Morelletti, those in the Gulf of Mexico. And yeah, we're expecting to have like a gradient of these genes in expressing these genes in the hybrids.
Okay, thank you very much. Super interesting.
Thank you.
Thank
We
you.
have time for one more question.
Thank you for your talk. Really interesting. I am just curious how the hybrids looks, the
No,
morphology.
yeah, I'm glad you asked that. They are an absolutely half mix. Morelletti is very robust, and the face is more like if you had hit him on the face, more rounded. And while the American crocodile is more slim and the face is really long, besides the differences in scales that are taxonomic diagnostic, but the hybrids are bigger, are the biggest of all, and they have like more, they're robust, but they're long but not as long. They are half mix. It's impressive how they look in the wild, but they are very big, and they are the hardest to manage. They're very strong.
Thank you.
You're welcome.
One very quick question. If there's any other, maybe from a trainee. Okay, if there are no more questions, let's thank our speaker again. Thank you for the very interesting presentation.
Thank you. See you.